Hello everyone and welcome to another day of high yield USMLE questions. Today we will be talking about biochemistry. So this is a 41st question of the medicalized question bank. So this is a biochemistry question and it's going to be more helpful for USMLE step 1 students. So step 1 students are going to be find it helpful. So we have a researcher. Researchers are studying the effects of fasting on cells. For this purpose, they expose the living cells to prolonged fasting. They found that these cells have decreased number of free ribosomes in the cytosol. So which of the following will be the effect of this finding? So now, for this, we need to step back and think about the function of the endoplasmic reticulum and the ribosomes. So now, if you have this, let's say this is the cell and this is the cell membrane. Inside the cell, this thing here, all the space is called the cytosol, and within the cell we also have different organelles. For example, we have, let's say this is the nucleus, and with the nucleus attached, just shooting off of it, is the endoplasmic reticulum, the rough endoplasmic reticulum. So now remember, so if you have a the ribosomes directly attached to the endoplasmic reticulum, that endoplasmic reticulum is called the rough endoplasmic reticulum. Then there are also ribosomes that are freely here available in the cytosol, in the cytoplasm. So these are called free ribosomes. These are called membrane-bound ribosomes. The ones that are attached to the endoplasmic reticulum are called membrane-bound ribosomes, and those which are free are obviously called the free ribosomes. Now, so now let's talk about what happens when they synthesize proteins. So remember, these free ribosomes, let's say this is a protein that this ribosome is synthesizing, this black. So this protein, let's say, is synthesized by the free ribosome, and the free ribosome is not attached to the endoplasmic reticulum. This is going to be synthesized, and eventually it's going to be left inside the cell. Now this protein can be used within the cell, not outside the cell. It's just going to be used within the cell. So the free ribosome synthesized a protein, and this protein is now going to be used inside the cell. For example, this protein can go to the mitochondria, it can go to the nucleus and be used, or it can just stay here as a cytosolic protein. So that's the free ribosome function. It produces proteins for the cell itself. It is not secreted. This protein does not go outside the cell. Now we have, let's say this is another ribosome here. This is another ribosome, and let's say this is synthesizing another protein. Let's say this blue is a protein that it synthesizes. Now on this protein we have something called the signal sequences. So now those proteins which have these signal sequences are going to be recognized. These sequences are going to be recognized by the ribosome, and what the ribosome is going to do to this protein is going to pull this protein towards the towards the endoplasmic reticulum. So this protein is going to be brought by the ribosome to the endoplasmic reticulum and into its lumen here, and this protein is going to start to get synthesized in the lumen of the endoplasmic reticulum. Also, this ribosome is going to come and attach here to this endoplasmic reticulum, and now this becomes a membrane-bound ribosome, and this endoplasmic reticulum becomes a rough endoplasmic reticulum. So remember, those proteins which has the signal sequences are gonna be synthesized in endoplasmic reticulum. Now, once they've gone through this endoplasmic reticulum, the next apparatus, the next organelle that they are going to go to is the Golgi apparatus, where it gets packaged, it undergoes some modification, and from the Golgi apparatus these proteins can go to the lysosome if it's a lysosomal protein, or they can go to the cell membrane, become cell membrane protein, or they can go outside the cell and secrete it. So remember, free ribosomes synthesize cells for the cell. They are not secreted outside the cell. The membrane-bound ribosomes synthesize cells in the endoplasmic reticulum lumen, and then those proteins go to the Golgi apparatus, and from the Golgi apparatus they can go to the cell membrane, the lysosome, or outside the cell. So the secretory proteins are actually synthesized by the membrane-bound ribosome. Now let's go to this question and see what's happening now. So now you will have a better understanding of this question. So this question is saying that we have decreased number of free ribosomes, just the free ribosome. They did not say anything about the membrane-bound ribosomes. So now, what is going to be decreased if you have decreased free ribosomes? Obviously, cytosolic protein. So this is the answer here. So now this wrong answer, this one wrong answer, the A, B, because these are extracellular proteins are synthesized by membrane-bound ribosomes. Cell membrane proteins and the cell membrane proteins are also synthesized by the membrane-bound ribosomes. So these two are synthesized by the membrane-bound ribosomes. Lipids are not synthesized by ribosomes. Ribosomes synthesize only the proteins. For lipids, remember the smooth endoplasmic reticulum. Hope this video was helpful, and if anyone likes the channel, they can subscribe and like the channel. They can also support the channel. It works. It helps the channel. If it you can support the channel, and the links will be in the description for supporting. Also, if anyone requires tutoring, they can email me here. Thank you for watching.